What is up, my Form 4s? Welcome back to our weekly lesson. This week, we are finally going into meiosis. This would be, I would say, a harder part of mitosis. Okay, I hope you're ready. I hope you got mitosis under your belt. Alright, we already have 2 lessons before. If you have not mastered them, kindly refer it back to my 2 previous videos. Alright, meiosis. What is this? Alright, last time we learned what is somatic cell. Okay, somatic cell means all the cells in your body. They will do mitosis. Alright, as for one type of cell in your body, which is the very special one, we call it reproductive cell. Alright, so only they will do mitosis. Okay, sorry, meiosis. My mistake. Alright, so only your reproductive cell will do meiosis. Okay, why is that so? Because as I said, you are diploid. Remember what is diploid? Your father is diploid, your mother is diploid. So in order to make you diploid, whatever, they need to only give you one set. Okay, they only need to give you one set, which is haploid. Haploid, you are diploid. It can't be your father does mitosis to produce diploid and your mom does mitosis and do another diploid. You will be tetraploid. That will be 4n. Okay, you'll be a monster, or not a human at least. Alright, so what is this process of changing diploid into haploid? That is what we call meiosis. Alright, meiosis. So you can safely say that your sperm and ovum, they are all haploid. Which means one set. How many chromosomes? One set. 23, which makes you 46. So next time when you want to have children, or you will give your children one set. Okay, your reproductive cell, which is your ovary or your testicle, they will do meiosis to produce gametes, okay, which are all haploid. Alright, so meiosis is a bit complicated because we have 2 parts here. We have meiosis 1 and meiosis 2. Okay, this video might be a bit long and I might split into 2 parts. Okay, depending on the situation. Okay, I will try to make this as slow as I can. Alright, so meiosis 1 has two parts. Sorry, meiosis has 2 parts. You have meiosis 1 and meiosis 2. Alright, so the basic concept is still the same. You have P, okay, pasang malam ada telephone, promats, okay, promats. So you have prophase 1, metaphase 1, anaphase 1, telophase 1, P1, A1, P1, M1, A1, T1, and then it will do meiosis 2, which is P2, M2, A2, T2. Okay, prophase 2, metaphase 2, anaphase 2, telophase 2. Okay, so they are 2 separated cell divisions, which means for meiosis 1, it will finish as 4 stages and then it will do cytokinesis to produce 2 daughter cells, which is similar to mitosis, and then it will do meiosis 2 again, which will produce another 4 daughter cells. Alright, I hope this makes sense. Alright, so there are 2 steps here. Okay, so what are the key points here? Okay, meiosis 2 is very simple. Meiosis 2 is basically like mitosis. You use sister chromatid. Okay, words like this, sister chromatid or chromosome. Okay, I hope you still remember them. Alright, for prophase 1, for meiosis 1 is our main point. Because we don't use sister chromatid, we don't use chromosome, we use a very special word, we call it homologous chromosome. Okay, you have a new word for meiosis 1, which is homologous chromosome. Alright, so let's look at prophase 1. Okay, so let's draw, shall we? Same like last time, let's draw. Okay, for prophase 1, okay, the basic prophase things still apply. Okay, still visible under light microscope, but this time it's not chromosome, it's homologous chromosome. Okay, chromosome. Okay, for all meiosis 1, P1, M1, A1, T1, or you need to use homologous chromosome. This is our keyword this time. Not chromosome, homologous chromosome. Alright, so homologous chromosome, visible under light microscope. Okay, same as last time. Only difference here, you have homologous chromosome. Okay, let's talk about homologous chromosome. You might ask, what the hell is this? Okay, homologous means two of the same. Homo means same, homosexual means same sex lah. Okay, so homologous chromosome looks like this. You will see this in the book. One is white, one is black, or other color lah. Okay, and then you will see something very funny. You will see this thing. Okay, this is what we call homologous chromosome. Okay, you might notice something. Hey sir, why is the leg one white, one black? Okay, so this place is called chiasmata. Okay, chiasmata. So the place is not so important. The process name, this process is what we call crossing over. Okay, crossing over. Why do we need to do crossing over? This crossing over has only one job, which is to create variation. How, you might ask? Okay, so when they change legs, they can change different sections of the legs. If you still remember, one chromosome has many genes here. You have maybe height, your body fat, your double eyelid, do you have ability to roll tongue, okay, your skin color and all. So they might change. So which will change with which, we don't know. That's why it's a very random stuff. That's why all your gametes, can you imagine, you produce, let's say guys, you produce I think few million sperms per day. All the few millions of them are different. Okay, can you create 5 millions of variation, let's say, different every day? They are all different. That's what makes it so amazing, because they are all different. Okay, the possibilities, how many trillions and trillions and trillions. Okay, so this is what's so amazing about this variation. So crossing over. Okay, crossing over. So when you draw them, make sure you make one leg black and one leg white. Okay, you don't need color pencil for this, just use your pen will do. Okay, so do you still remember our mitosis has 2 points? Okay, chromosome visible under light microscope, nuclear envelope disintegrate. So same thing here. Okay, nuclear envelope or nuclear membrane disintegrate. So we have one more point here which has crossing over. Okay, crossing over. So that's the special part. So prophase 1 is especially important because you have one very special word here, which is crossing over. There's another name for it. Some books, they'll write synapses. Okay, I wouldn't bother. I will just write crossing over. Okay, either case is fine. Okay, so if you draw it in your notes, it should look like this. Okay, you should draw two lah. Okay, one black, okay, with white leg, and then one white with black leg. Okay, and then I will draw another one. Okay, just for illustration purpose lah. Okay, black, okay, and white. Okay, so this is what we call crossing over. Alright, so for metaphase 1, anaphase 1, telophase 1, okay, basically everything is the same. Okay, only difference here is this time we don't use sister chromatid, we use homologous chromosome. Okay, so when you draw metaphase 1, it looks like this. Okay, please make sure your legs remain. Okay, you draw out the legs. Okay, I will draw two lah. Let's not waste too much of time drawing here. Okay. Alright. Okay, so what do you write here? Do you remember what happens in your metaphase in mitosis? Okay. Sister chromatid arranged at the, or you can say chromosome arranged at equator. Remember your equator? Okay. This time we don't use chromosome, we use homologous chromosome. Okay, change all your chromosome in your mitosis to homologous chromosome, then it will be fine. So homologous chromosome arranged, okay, at equator. And this, you still remember what happens? okay, the spindle fiber is attached at the centromere. Okay, so I'll just write spindle fiber attached, okay, at centromere. All right, so what happens at anaphase 1? Same thing. Okay, I wouldn't draw the color one lah, you draw it yourself. Okay, so in your metaphase, in your mitosis anaphase 1, in anaphase, there's no anaphase 1 in mitosis. So what happens? You say sister chromatid separate. So now it's the same thing, but we don't use sister chromatid, we use another word, which is homologous chromosome. Okay, separated. Okay, move towards opposite pole. Okay, move towards opposite pole. All right, so what happens at telophase? Same thing, homologous chromosome reaches opposite pole. Okay, make sure you draw your color legs. Okay, the color, you draw it yourself. I'm just gonna keep my time short here. So homologous chromosome reaches opposite pole. All right, so as you can see, nothing difficult here. It might be tricky. So the only tricky part here is prophase 1. That's why I put a lot of focus on prophase 1, because there's a lot of keywords to memorize. You have synapses, you have crossing over, you have chiasmata. Any else word? Okay, later I'll talk about the notes. Okay, you might ask, sir, where is meiosis 2? Okay, meiosis 2 is exactly the same as mitosis. Whatever you write at mitosis, you can write for meiosis 2. Same thing. What happens at prophase 2? Okay, chromosome arranged at equator. What happens at metaphase 2? That one is so visible under light microscope just now. Okay, sorry. Okay, arrange at equator. And then what happens at anaphase 2? Sister chromatid. Okay, you can use back sister chromatid this time. Okay, sister chromatid separate. And then what happens at telophase 2? Sister chromatid reaches opposite pole. Okay, so meiosis 2 is exactly the same as meiosis. Okay, so I wouldn't put too much focus on meiosis 2. Okay, because since it's almost the same as mitosis, they don't like to ask this in exam. Even though, yes, it's very rare. Okay, why? Because they give you free marks like that, right? Okay, so let's look at the points. All right, so prophase 1 again is my focus. So can you see the crossing over here? Okay, they didn't change leg first. Okay, this is what happens here. All right, so let's look at the point. Okay, visible. Okay, same thing. And then homologous chromosome. They form pairs of tetrad bivalent. Okay, what is tetrad bivalent? This means the shape. Wow, we have so many keywords, isn't it? And then synapsis, crossing over, chiasmata. Okay, I'll explain this. Okay, chiasmata is the place. Okay, crossing over is the process. Tetrad is the shape, or bivalent is the shape. Okay, so don't confuse yourself. So you have so many keywords here. You have 1, 2, 3, 4, 5. You have 5 keywords here, so don't confuse yourself. Okay, I would say the most important is the crossing over lah. Okay, crossing over, crossing over, crossing over. I cannot stress this enough. This is the most important point here. All right, so what is this for? Like I said just now, it's to make genetic variation, to make sure all gametes are different. All gametes different means all of you are different. If you were to give birth to 1 million offspring, I don't think you can. Even 100 is like a challenge really. I don't think 100 is even 20 is a challenge. Would you have 20 children? So let's say if you live until 1000 years old, you have 1 million offspring. All of them will be different. There will not be 2 of the same people. Isn't this amazing? Okay, historically speaking, okay, so many human beings have come and go. So many organisms, we talk about all organisms have come and go. No 2 organisms are the same. Besides, of course, the cloning I talked about last week. Okay, this is what makes cloning so amazing, because that's the first time in history 2 individuals are the same. Okay, impossible. Okay, this is just nature. Okay, this is what's so amazing about this. All right, so let's talk about metaphase 1. Okay, homologous chromosome arranged at equator. You see, same thing. Just we changed the chromosome last time to homologous chromosome. Okay, spindle fiber attached to centromere, same point again. And then what happens? Okay, spindle fiber contract pull, you don't need to know this. Okay, pull to opposite pole. Okay, this is just a clear diagram for you. Okay, doesn't matter. Okay, so haploid number doesn't matter. Okay, they reach opposite pole. Okay, and then they will do meiosis 2. Like I said, meiosis 2 is all exactly the same as mitosis. Okay, so what happens here? Okay, tightly coiled. Okay, visible under light microscope. Okay, nuclear membrane disintegrate, same thing. Okay, chromosome align the equator. Can you see? Just now we use homologous chromosome, but here same as mitosis, we use back chromosome. Okay, attached to centromere. And then what happens? Sister chromatid divides. See, we can use back the mitosis term. Okay, sister chromatid. Okay, so move to opposite pole. Okay, here's what you need to take note when you're drawing. Can you see the leg? The color is still there. Okay, so don't ignore. If this is meiosis 2, this is what you need to take note. Yes, it's almost the same as mitosis, but you still cannot ignore the color of the leg. Alright, and then telophase 2 reaches opposite pole. So simple. Okay, why do I say it's difficult? I just want to scare you so that you can pay attention. Okay, what's so difficult about meiosis? That's why I said mitosis is the foundation, is the basic. If you suck at mitosis, which I don't understand why I already repeated so many times, then you are gonna fail your meiosis. Alright, so damn simple. For me, this is even simpler than the previous chapters. Why? Because you have so many sugar to memorize. This one nothing to memorize. It's all common sense. It's a processed stuff. It's not dead stuff like types of sugar or protein or whatsoever. Alright, let's look at exercise. Okay, so there will be a part 2 meiosis video. Okay, I will sum this chapter up in the next video. Alright, so we'll do some exercise today. Alright, so this is a particular stage in meiosis. So what stage is this? So they are all already, you see they are changing legs. This is prophase 1. If you don't write the 1, it's wrong. Okay, bio is very specific. Describe the behavior during the stage shown. Okay, what are they? So you can say visible under light microscope. Okay, so if you say, sir, die lah this time, I forgot about prophase 1 already, then at least you can write some mitosis stuff. You are still correct. Visible under light microscope. What else? Okay, nuclear membrane disintegrate. You see, same thing. Okay, if you say, oh shit lah, I forgot crossing over that word, then you don't write lah. Write the other 2 points lah. Okay, so crossing over. Okay, if you are the overachieving type, you can write all the 5. Okay, they form tetrad bivalent. They do crossing over synapses at the chiasmata. Wow, bravo, good for you. Okay, so let me clap for you. Okay, because you are very hardworking. Okay, but kind of a show off if you ask me. Alright, so B, name point X and process takes place at X. So what's the place called? Okay, it's called chiasmata. Okay, so what is the process called? Okay, crossing over, or you can say synapses. Okay, C, explain why is this important. Okay, for genetic variation. Okay, what else? Okay, you can even say to produce haploid gamete. Okay, we need to make sure the gametes are haploid, means one set. That's why we do meiosis. Alright, so name and explain chromosomal behavior after this stage, which is metaphase 1 lah. Okay, so what happens? So the homologous chromosome arrange at equator. So what is attached to what? Okay, your spindle fiber attached to centromere. Hey, so easy lah. Okay, so draw anaphase 1. Okay, so anaphase 1 looks like this. Okay, and then your homologous chromosome, they are pulled to opposite pole. Okay, so you draw the other side lah, same thing lah. Okay, so based on the diagram, what is the characteristic of this stage? So what happens? Homologous chromosome move towards opposite pole. Okay, what causes chromosome to act stated in G? Okay, what causes this? Okay, where is G? E. Oh, I think this is stated in G. Okay, whatever. So what causes chromosome to act this way? Okay, the shortening. Okay, the shortening of your spindle fiber. Okay, it shortens. Okay, it pulls them apart. Okay, name structure G. What the hell is G? Yes, G. That's the centriole. Okay, only in animals, plants, they don't have centriole. How is the formation of gametes affected if G fails to function during meiosis? Okay, so if there's no centriole, there will be no spindle fiber. So no spindle fiber, how? Homologous chromosome fail to separate. So what fail to separate, how? One cell would have all the chromosome, one cell will be empty. Okay, might sound funny to you, but that's this is a very common question. Okay, they like to ask this type of question, some sort of like the K-but thing. Okay, draw two daughter cells as the result of C2. So where is C2? C2 is basically they want the meiosis 1 lah. After meiosis 1, what happens? You have one side of this. Okay, no more homologous chromosome. Okay, because they separated already. Okay, so make sure you color them. Okay, let's say this is black and white. This is white and black. Okay, black. Oh, if this is black, then this should be white. Sorry. Okay, because they are the pair. Okay, white, black. So this is white, so this is black. Okay, so make sure you label. Okay, so this is your nuclear envelope or nuclear membrane. Okay, chromosome. Okay, so that will be it. Okay, so 4 stages of cell division. What is this type of cell division? Okay, whenever you see there's color change, this is confirmed meiosis. Okay, correct sequence. So where's the prophase? Where's the prophase? Okay, this one they jumpo. Okay, this is, let's label them. What is this? This is telophase 2. Okay, this is T2. How would I know? Because there are only sister chromatid left over. There's no more chromosome, means it's telophase 2. And then they already reached opposite pole, confirm this is telophase. Okay, how about this? This is anaphase 2. Okay, how would I know? Again, the sister chromatid already separate. There's no homologous chromosome here. So this is anaphase 2 and they are separated, moving towards opposite pole. What about this? This is metaphase 1. Homologous chromosome is still here and they are arranged at equator. I hope this makes sense. Okay, how about this? I think this is telophase 1. Okay, how would I know? Because almost the same as this, only case here, I still have my chromosome. Okay, it means they already done the first part. The chromosome already separated. They still haven't separated the sister chromatid. Okay, I hope this makes sense. All right, so who does what first? I think X happens first, meiosis 1 first. Okay, and then Z, telophase 1. Next would be Y, anaphase 2. And then next would be W, telophase 2. Okay, these questions can happen. They don't give you all the 8 stages. Okay, PMAT 1 and 2. No, they give you like any of the 4. Okay, they want you to guess yourself. Okay, this takes a bit of identification work. So like I said, for me, I will write this down. Easy for you to refer. Alright, so B1. What happens to the chromosome during stage X? So stage X here. What happens to the chromosome? So they are arranged at the equator. Okay, I've already repeated this like for a million times. If you still cannot understand, please pay attention. Okay, you have been locked at home for too long. Your brain is getting rusty. Go and do some exercise. Okay, you can do exercise at home. I'm doing exercise at home. Okay, to keep myself sane. If not, I'm going crazy. Okay, go and sweat a little bit. Okay, open some YouTube videos and search for cardio at home. Okay, sweat it out. You need sweat to activate your brain. Okay, exercise is more important than you know. Okay, so number 2. What happens to the chromosome after stage X? And why is chromosome behavior important in this cell division? So after X is meiosis 1. After meiosis 1, we do anaphase 1. But they don't want the name. They want what happens. So what happens at anaphase 1? Okay, homologous chromosome. I'll just write short form lah. Homologous chromosome separate and move towards opposite pole. Okay, so that's the first point. Why is this chromosomal behavior important? Why do we need to separate them? Okay, to ensure daughter cells are haploid. Okay, we need to make sure your gametes are haploid. If we don't do this, your cells would be all diploid, all your sperm or ovum will be diploid, and then if you happen to marry another guy like that, your son will be tetraploid. Okay, he could be a banana or a potato. Okay, more chromosome doesn't mean better. All right, so C, fusion of sperm with ovum and process P. So what is process P and Q? This is basically Form 3 stuff. So what is P? Okay, so what is process P? I think P should be fertilization. This is, I don't think it's Form 3, this is Form 1 stuff, isn't it? New syllabus. So fertilization. So fertilization will produce something called Q. So what is Q? You start from that one cell, which is zygote. Okay, and then zygote will do mitosis. I'll finish this to form fetus. Okay, fetus will do mitosis to form you. All right, so this is not chapter 6 stuff. This is Form 1. All right, so this is a karyotype. Oh, genetic disease. I'll talk about this next week. Okay, we still have another lesson go on. Okay, so comparison. Okay, so if you notice here, this is mitosis. Why? Because there's no leg color change here. Okay, and then this is meiosis. How would I know? Because there's color change and then there's crossing over. So obvious. Identify X and Y. Okay, so they want cell division X. So X is mitosis. Y is meiosis. Ayo, like that also can ask. Like that also got 2 marks. Okay, so good. Choose the cell division that results in genetic recombination. Genetic recombination means I'm changing. Whenever you see genetic, confirm this is Y. Of course, got crossing over, ma. Explain your choice or answer. Okay, so what happens here? Okay, crossing over. Okay, or synapses. Okay, so what happens here? Exchange of genetic material. All right, so P and Q are stages in X and Y respectively. Describe the behavior of P and Q. So what is P? What is P? They skip a stage here. So this is P1, then Q, then this is A1. So what happens here? This is M1. So this is P, M, this is T, and then this is A. Basically, P is anaphase, Q is metaphase 1. So describe the behavior of chromosome in P and Q. So simple. So what happens at anaphase? Okay, so chromosome, not chromosome, sister chromatid. Okay, separate. Okay, move towards opposite pole. Okay, so how about Q? Q is metaphase 1. What happens at metaphase 1? This time we cannot use sister chromatid. You must use homologous chromosome arranged at equator. Okay, as you can see, the questions are pretty much repetitive. Okay, so if you finish this thing, you should be able to score an exam. Okay, so let's discuss one more. Okay, suggest why shows the failure of chromosome 21. I'll talk about this next week. This is genetic disease. This is what we call Down syndrome. Okay, have you heard of the disease Down syndrome? Okay, I'll talk about this next week. All right. So let's look at this. Between P and Q, which undergoes meiosis? So which one has crossing over? Of course, Q lah. 2 reasons for your answer. Okay, formation of, what is this? This is called homologous chromosome. Okay, what takes place here? Crossing over. Okay, so what happened? This. If you can't even draw this, what the hell are you learning? Okay, so good for you is this part is very common in exam. Okay, and there's not much K-but thing here. Complete the diagram above 1 mark. Explain how plays an important role in producing variation. So how does this produce genetic variation? So you can say exchange of genetic material. Okay, So there's not much to talk about here actually. How do you get the 4 marks? Okay, exchanging of genetic material. Okay, sometimes even you say via crossing over, you can get another mark. Okay, via crossing over, or you can even say maintain haploid number. Okay, things like that. Okay, the 4 marks are easier than you think sometimes. Alright, C. Event of mitosis in animal plants are very similar with the exception of absence of centrioles in plants. How plant undergo mitosis and the absence of centriole? Not gonna talk about this. Okay, not so important. Oh, basically this is next week's stuff. Never mind, we shall skip along. Okay, so I think that will be it for this week. Okay, easier than you think, isn't it? Okay, like I said, if you still have doubt about mitosis, kindly refer back to the 2 videos I have discussed the previous 2 weeks. How many did I make? 2, 2, 2. Not 1, not 3. I could have made 3, but you'll be bored. Okay, I made 2. Okay, 1 I talk about how to draw, the other one I talk about it's actually 3 though. One of it is exercise. Okay, there you go. You see, I've made so many videos about mitosis. So kindly refer it back. Okay, get it under your belt, get it familiar, then only you start this. It's not as hard as you think. Okay, and make sure you do exercise. If you learn but you don't do exercise, there's no point for you in watching this video. Okay, you might think, oh, I got this. Okay, no, you don't. You need to do exercise to reinforce the memory into you. Okay, all right, enough babbling from me. Okay, there I go again. Okay, so next week I will finish this chapter. Actually, according to syllabus, we should finish this by this week. But no matter, we are taking this slow and steady, and we will win the game. All right. So there's no need for you to rush because just do your part during this holiday, and when you go back to school, you can just sit and chill. Whatever the teacher says, just sit there. I've already learned this. Okay, no matter, she's gonna rush, I guarantee it. Okay, and when you see people climb cupboard here and there, you just chill because you have already learned it. All right, so please, your future self will thank you for spending this 1 hour or so revising it every week. Okay, just 1 or 2 hours per week is more than enough for you to learn something new. All right, so I hope you learned something today. Okay, I will see you again next week. Hopefully the MCU will not be extended. Okay, we are all going nuts, I believe. All right, so see you again next week. Take care and goodbye.